Hello students, as we are doing chapter 5 and we are discussing about the cell organelles, so now it's the turn of ribosomes. So ribosomes they were first observed by George Pallet in 1953. George Pallet observed these structures as some granular structures present inside the cytoplasm, and first time they were observed in an eukaryotic cell. Fine. And if we come to the composition of ribosome, then ribosomes are generally composed of RNA and proteins. So main genetic material of ribosome is RNA. And the most important property about the ribosomes, about which sometimes question is also framed in the entrance exam, so you should focus here that these ribosomes, they are devoid of membrane. They do not have any kind of membranes around them. Okay, they lack membrane. Now if we come to the types of ribosomes, there are two main kinds of ribosomes, ATS and 70S. ATS ribosome is of eukaryotic cell and the 70S is of prokaryotic cell. Here this S unit is Swedberg's unit, which stands for the sedimentation coefficient, or you should say that it's a measuring unit for density and size. Means like we weigh weight of a body in kg or gram, the same way if we come to the if it comes to the measurement of the size of ribosome, then it is measured in Swedberg's unit or sedimentation coefficient. Fine. So coming to the structure of ribosome, ribosome is a quite interesting structure because each ribosome is composed of two subunits. You can see from this figure that I have drawn both the structures of ATS and the 70S too. So you can see that each ribosome is composed of two subunits. This bigger one is the larger subunit and the smaller subunit. Same here in 70S2, there is one larger subunit and one smaller subunit. Fine. So if we come to the eukaryotic ribosomes of ATS size, these two units, you can see that the larger subunit is of 60S and smaller is of 40S. But if you will add these two, 100S must come. But when these units combine at the time of protein synthesis, then some of the portion of these ribosomes, they get overlapped together. So because of overlapping only, size get reduced to 80S. Same here, you can see I have written here the larger subunit of a prokaryotic ribosome is of 50S and the smaller is of 30S. So at the time of protein synthesis, when they combine together, when they associate together, then the size get reduced to 70S. Although if we will add these two units, it should come 80S, but it is not 80 exactly, it is 70S. Because of the very small overlapped portion, the size get reduced to 70S. Fine. So you can see that each the ribosomes, they are made up of two subunits. But when these subunits are not involved in any of the specific function, means when these are not involved in any specific function, they remain separate from each other. But when they are ready to perform a specific function, like protein synthesis is the main function of these ribosomes, so at that time they come together and then they form an associate complex. Fine. Now coming to the functions of ribosomes, there is a main function of ribosome which is protein synthesis. Fine. This is the only main function of ribosomes. At the time of protein synthesis, different types of ribosomes present inside cell, they come together and then they perform this function. Now I am coming to the next structure of next organelle of a cell, that is a cytoskeleton, which forms the cytoskeleton. As the name indicate, cytoskeleton means the structures which will form the skeleton of a cell. Okay. So these cytoskeleton, they form an elaborate network of filamentous proteinaceous structures. Means they have several proteinaceous filaments. Okay, they are also present inside a cell, and they are present in cytoplasm of a cell. So all these proteinaceous filamentous structure will constitute cytoskeleton. What can be present? What are the different types of these filamentous tubes? These filamentous tubes can be microtubule, they can be microfilament, and they can be intermediate filament. Fine. These are the three different types of proteinaceous filaments present in the cytoplasm of a cell. Now, what is the function of these cytoskeletal structures? You can see I have written here, these are the main functions. As being proteinaceous, they will be little rigid in the structure and they provide the mechanical support. Fine. Now, if you discuss about the proteins, then the proteins are most of the proteins actually, they are contractile in nature, means they help in the motility of the cell. Same here, as these cytoskeletal structures are generally proteinaceous filaments, so that's why they will also help in the motility of the cell. Now, the last function I have noted here, they also help in the maintenance of the shape of the cell. So definitely, if they are providing mechanical support, if they are helping in motility, it means somewhere being little rigid, they will also help in maintaining the shape of the cell. Fine. So these are the three main functions of cytoskeleton. Now I am coming to the next structure of a cell, which is cilia and flagella. And I think most of you must have heard about these two. These two are actually, they help in the movement of a cell or the medium around the cell. So you can see it's a very nicely described in your book, so I'll take the help of your book. So look here, you can see here internal structure of cilia and flagella they have shown. So you can see that these structures are covered by an outermost structure which is membrane. You can see here it's written plasma membrane, the outermost structure of cilia and flagella. Now the whole structure left is called as axoneme. Okay, the core of the flagella and cilia is called as axoneme. This axoneme term is given here. This is axoneme. Fine. So what is present in this axoneme? This axoneme is here important to describe. So you can see that in the axoneme there are several radially arranged microtubules are present. You can see 1, 2, 3, 4, 5, 6, 7, 8, 9. These are the 9 doublets, because in each there are 2 microtubules present. So there are 9 doublets peripherally arranged. They are arranged at the periphery. Okay. And in the center, two uh, two microtubules are arranged, and these two are connected through a bridge. Okay. And these centrally located two microtubules, they are covered by a membrane too, which is called as central sheath. Fine. So, let us recall the points which we have discussed about it. Outermost structure of cilia and flagella is plasma membrane. The whole left portion is called as axoneme. This axoneme is consisting 9 doublets of radially arranged peripheral microtubule and a pair of centrally located microtubules. Okay. So, this arrangement is also called as 9+2 arrangement of microtubule. So, generally in cilia and flagella, this 9+2 arrangement of tubule is seen. This is also called as 9+2 array. 9+2 array. So, you can see that these two centrally located tubules, they are covered by a central sheath. Okay. They are connected by a bridge. And now the important point to note that from each doublet, one from each doublet of radially arranged microtubule, one from each pair, like this one from this pair, you can see this one from this pair, one of the microtubule from each pair is connected to this central sheath. You can see these spock kind of structures, दोनों में से एक से निकल रहा है. So, this is called as radial spock. Radial spock. So, all these microtubule are connected to the central sheath with radial spock. Fine. Thus, there are 9 radial spocks, and the peripheral doublets, they are also interconnected by the linkers. You can see these are the linkers. These are also interconnected. ये आपस में भी जुड़ी हुई हैं through these interdoublet bridges. You can see these are the linkers. Okay. So, you can see that the whole structure is interconnected to means it will function as a whole. That is why it is helpful in movement of the cell. Okay. So, these cilium and flagella, they actually emerge from centriole like structure, which is called the basal body. That basal body is present inside the cell, and that basal body will give rise to cilia or flagella. How? Like if it is the body of Euglena. Okay. So, the point from where this cilia or the flagella arises, this is called as basal body. Basal body. Fine. Basal body. So, main function of cilia and flagella is only the movement actually, that is responsible for cell movement specifically. So, you should know that the prokaryotic bacteria, and they also have flagella, but they are structurally different from that of eukaryotic flagella. Okay. And I think you must have seen the paramecium figure. Paramecium also bear cilia. So, these are the specific organelles which are helpful in the movement of the cell. Fine. So, today we have covered, we have started from ribosomes, then we have discussed little bit about cytoskeleton, and then we have discussed about the cilia and flagella. So, this way you can make points, and then only you can understand. Even if you understood this figure, then you can frame your own points. No need to cram everything. Fine. I hope you understood this topic. Thank you.